Hello, everybody. Good morning and also good afternoon if you're joining us from Europe or Asia. I'm Abigail Chu, Chief Scientific Officer at Precisionary Instruments, and I'm delighted to welcome all of you to today's Precisionary Scientific Series webinar. It's my pleasure to introduce today's speaker, Dr. Régis Joulia, an advanced research fellow and Wellcome trust-funded group leader at the National Heart and Lung Institute, so NHLI. Dr. Joulia's research focuses on understanding how immune cell activation reshapes the lung during respiratory disease, particularly how inflammation influences blood vessel organization and tissue remodeling. So don't forget about the blood vessels in the lung when you talk about respiratory disease. His laboratory uses precision cut lung slices, or PCLS, to study the immune cell behavior within intact lung tissue. And that provides a really powerful three-dimensional platform for investigating allergic and inflammatory responses. So today I want to welcome Dr. Joulia here to share how his team uses PCLS to visualize the immune cell distribution in the lung, discuss the advantages of this approach over traditional tissue sectioning methods. Of course, we're a big fan of PCLS at Precisionary, and also explore future applications for lung and mucosal immune response research. We're very excited to have him with us today. So please join me in welcoming Dr. Régis Joulia. And what I wanted to add is, for those of you attending our webinar today, don't hesitate to enter your questions and comments during the talk, during Dr. Joulia's talk in the chat. I'll be monitoring it. And at the end, there will be a moderated Q&A session. So feel free to also show yourself and ask questions, but I'll also be keeping my eye on the chat box. So with that, please, let's welcome Dr. Joulia.
Hello, Abby. And thank you very much for the invitation to present today. Let me just share my screen now. Oh, just disappeared. No. Oh, 1 minute. That's it. Okay, so yeah, I would like to thank again you for the invitation to speak today at this event. I think it's a technology that we really enjoy in the lab to do, and how we're going to use PCLS to actually do this. So yes, my lab is working at Imperial College, where the National Heart and Lung Institute, and we are using PCLS approaches to better understand basically the lung architecture in 3 dimensions and how this is changing during disease. So my lab specifically is interested into the lower airway and the disease associated to this lower airway. So if you're not very familiar with the lung anatomy, the lower airway starts with the trachea. And following the trachea, you have this area called bronchi or bronchovascular space. Just get rid of it. And bronchi or large airway space, which is very interesting for us because it's really a starting point of the disease. I'm just going to explain just after. And finally, you have the respiratory zone, the alveoli, where the gas exchange occurs. And along all of this track, there is really a highly stratification of both cells, immune reaction, type of disease encounter, etc. So the lung is a very well organized and defined structure, but sometimes I think we consider it as a single entity and we look at lungs, but I think we really need to look into detail and how this is really changing according to the anatomical structure. Today, I'm going to mainly speak about this inflamed airway space. So this is really the starting of the branching point of the airway. And why we're interested in this area is because it's really, we call it a crossroad of immune and stromal cells. Locally, there is large airways where the external threat will come in. So here comes the allergens, viruses, pollutants, etc. And these irritants are going to be directly exposed to the surface of the epithelium. And just beneath this epithelium, you are going to find a whole range of resident immune cells, what we are interested in the lab, such as macrophages, dendritic cell, mast cell, but also adaptive immunity. We know that now this is one of the primary points of generating immune response against this external threat. So you find T-resident memory cells, etc. But also non-structural cells that are important to generate immune response, such as neuronal projection and a very vast different range of blood vessels. And these blood vessels are very interesting for us because we know they are present there in this region, but we don't really know how they're impacting in the context of disease. And I will go back to this blood vessel in a minute, but the disease that we are particularly interested, and I will go into speak about in this presentation, is asthma. So why looking at asthma is important? It's a disease that is impacting millions of people worldwide, about 300 million, and about 8 million people in the United Kingdom. But that's a disease that is currently incurable. All the treatments are basically alleviating symptoms, preventing asthma exacerbation, but they are not actually curing the disease. One other critical aspect of this disease, it's about nicely estimated that about 10% of children worldwide are impacted by a form of asthma, and this is increasing every year. We still don't understand the origin of the disease. We know it's multi-combination of factors that are both from genetic and the environment, but we all know that dysregulated immune response and tissue remodeling are key factors. So we believe that this early allergen exposure during this period of growing up, in early life, is driving this sort of aberrant immune response, and after leading to this, a lot of work from different labs, including the lab from Bart Lambrecht, showing how these allergens are exposed to dendritic cells, deriving a type 2 immunity response, eosinophilia, mast cell activation, basophil recruitment, that are going to lead to the asthma symptoms. Due to the chronicity of the disease and the fact that these children and adults have asthma for years without any kind of curative treatment, the tissue starts to be really highly remodeled. And you have here one of the images that most asthma community is using of a normal airway bronchovascular space, where you can find, as I said, the epithelium, large blood vessel next to it, and the alveoli structure that are surrounding this sort of bronchovascular space. And during asthma, this tissue has been really dramatically changed. The epithelium is highly remodeled, the smooth muscle is highly also more thicker, and around this now airway, whereas you could see before blood vessels and alveoli structure, now you can see completely the absence or completely change of structure of this also blood vessel organization. And this is what my lab is looking and interested in understanding the mechanism of how we can go from normal to this asthma structure. Why looking also as well at blood vessel in asthma and other respiratory diseases? Because patients with asthma, and this is also very well described for COPD, have a very high increased risk of cardiovascular disorder. So they have an increased chance to develop here, for example, heart failure. That was a large study performed in Denmark showing that patients with asthma with early or late onset have a significant increased risk of heart failure later in life. And for COPD, risks have already been described for decades, but we still don't really know the mechanism. What is also quite dramatic is that when we looked at the survival of this patient following any kind of vascular disorder, which could be heart failure, but also stroke, cerebrovascular disease, or another peripheral vascular disease event, patients with asthma, which are here indicated in red, are doing worse following this event, indicating that they do have this sort of like less stable vasculature, which has been that when an event is going to occur, which is maybe not related even to asthma, these patients are going to do worse, indicating that they have like some kind of more fragile, difficult vasculature, which is able to sustain this sort of serious injury. And we believe that the lung is really a starting point of this disease, as I mentioned, but also really where we're trying to understand is these airways are a starting point of this disease. And we believe that the immune cell and the interaction that we have locally are going to be critical in this interaction. And the lab is interested into mast cell and the relationship with the other structural cell of the blood vessels, namely the endothelial cell and parasites. I'm just going to mention one slide on parasites because people are sometimes not familiar with this cell. They are very complex to study, and this is where PCLS has been essential for us to better understand them, because they are parasites, they are surrounding different type of blood vessels, capillary and postcapillary venules, and they have this sort of very strong association with endothelial cell, and they are critical for blood vessel maintenance. They secrete the factor that allows endothelial cell to grow, but they can't be extracted from the lungs. They are very fragile cells. They are very large also as well, which makes them really difficult to use flow cytometry or other type of fluid-based technology to isolate them. And they have this sort of, as I mentioned, this sort of strong interaction with endothelial cell. There are reporter mice that are being developed now, looking at where they are present in the lungs, and this was one of studies that also used PCLS at the time, even if they don't call this, but from the lab of Ralph Adams, showing here the red cells, which are the parasite, and they have this sort of like type of neuronal structure where they have a large cell body, and after projection that are going against the endothelial cell. And mast cells, which are present next to this parasite, are also very intriguing cells. They have historical association with blood vessels, and this is what we have done before. We have looked into before this mast cell in all different tissue, but here I'm going to mention their role in asthma, and now we use PCLS to study them. They have this sort of historical role in allergy and asthma because they secrete histamine. They are the main cell secreting histamine. And I'm sure most of you actually have known what antihistamine can achieve now for patients with allergy, which have been quite a great relief for a lot of people. The mast cells are really this sort of classic resident immune cell. They can respond to almost any kind of stimulation, including IgE-mediated stimulation. They express all types of TLR and neuropeptide. And in response to these different stimuli, they can secrete they can release their granules, because they have actually a massive store of these intracellular granules. And these granules contain a lot of proteases, cytokines such as TNF or interleukin-6, and finally histamine, which is maybe the most famous mediator that mast cell can secrete. They are present very, very early on during development. We still don't really know why at embryonic stage they are required, but they are required. And they are present both in mouse and human, but I think this is still currently investigated. But the goal is really to see that they are present already very early during life, and they could be imprinted very early by allergen presence. So this is what we are doing. This is the objective of the lab overall, and to better understand the impact of chronic lung disease on the vasculature, and looking at this using this sort of vascular unit using both mouse model and human model and PCLS all across these two different organisms. And we use imaging to analyze this PCLS, and also we use now also full organ imaging. The PCLS is our day-to-day experiment lab. And if you are not very familiar with this sort of slices, we use the vibratome, which is we really like the one from Precisionary instrument, and we use this sort of like lung slices, which are about 200 micrometer in terms of thickness. And when we do this in mouse samples, we do after this sort of live section that we can image using confocal or light sheet microscope. And you have here an image of a video of a PCLS where the vasculature is stained in green. The airways are stained in blue, which is using smooth muscle. And finally, you have mast cell, which is a cell I mentioned about, which are purple cells. And these purple cells are present all across the side of the lungs, but they're also present in this sort of bronchovascular space in mice. And this is a video is going to zoom in now. So you have here a large airway, and you're going to see now a bit better. So you have the airway and the smooth muscle surrounding it. And you have the mast cells which are present there, that are these purple surface cells, and they are completely at the junction between this airway there in the middle, and you have here a blood vessel which is just lining up these airways. And this is what we call the bronchovascular space unit, and this is where you're going to see when we inject allergen, a lot of immune cells are recruited in this area. And as I mentioned, so we use a Precisionary instrument to use this PCLS, which is really a great tool for us because we can generate a lot of sections from a single animal, and they are very stable across time. We can image them for quite a long time. For this presentation, I'm just going to show you what we have done with fixed PCLS, but we can also do live PCLS imaging, which is really also a super interesting tool. So in this first project I'm going to mention to you, we looked into the impact of allergen during early life and used PCLS to track immune cell activation and how mast cell could influence tissue and structurally. To do this, we use biopsy mice that we expose to allergen from day 7 of life, and we expose them 3 times a week for a period of a minimum 3 weeks, which allows us to basically analyze how the development of the asthma is working. We also stop the allergen treatment, and we see after if the inflammation can resolve by itself, but also what are the permanent structural elements that are changing, that are still maintained across time during the resolution phase. And here you have an image, another video of the PCLS where we analyze the lung vasculature only. And you have, again, the CD31 staining for endothelial cell for the blood vessel in green. And you have this area, which is this bronchovascular space. And here you have here an airway here in the middle. And on top of this, you have these 2 large blood vessels and the adventitial area, which is just surrounding this area. And here you have now the parasite staining, which is in purple for PDGFR beta. And you can really appreciate how how there is but like thousands of parasites in these lungs. And that really, we believe that they play a critical role during the development of asthma. So we use this sort of PCLS platform to analyze multiple elements using imaging tools such as Imaris. So we can analyze how endothelial cells are covering the PCLS and how basically the volume of endothelial cell within the image. We can analyze vessel density. We can analyze cell number, such as parasite cell number, which we can do. Parasite coverage, and I'm going to show you this after what I mean by this. But here I want to explain that basically endothelial cell and parasite have this strong interaction. So we are trying to understand how parasites are covering this endothelial cell. And we can analyze the distance between this endothelial cell and parasite, and finally the infiltration of immune cells using CD45 here as a marker. So when we looked at this parasite, as you saw from the video, there was like about hundreds of them that are highly abundant across the adventitial area. And when we zoom in, and what do I mean earlier by parasite cell coverage? So you have here zoom-in images using again here of a PCLS of 3 small airways and 2 blood vessels adjacent to it. If you look at the PDGFR beta staining and we zoom in, we see this nice parasite here in the middle where we can see its cell body and the cell protrusion that are lining up and just in connection with the endothelial cell. So this is really a critical parameter for parasite because they use this extension to be maintaining endothelial cell fitness. They also secrete a lot of extracellular matrix, so they can also secrete this extracellular matrix around blood vessels to maintain them, and they can also secrete angiogenic factor. This loss of protrusion is a very critical marker, which is now super well acknowledged in the brain, where one of the early events happening in the during vascular dementia and Alzheimer is this loss of parasite protrusion. Why we are losing protrusion, we are still not exactly sure what's happening in the brain, but we believe that this may be an impact of early inflammation. And we are currently looking into this, if this phenomenon could be also similar in the lungs during chronic lung disease. And this is what we did. So we analyzed across the mouse model, across these different time points during the development of asthma in the resolution phase, how parasite and endothelial cell were speaking to each other using PCLS and 3D quantitative imaging. You have here an example of a PVS-treated mice where you can nicely see the vasculature next to this airway, which is everything looks fine. You have the alveoli structure. The bronchial vessels are just lining up these airways, and here in the middle you have air. When we look at the parasite network, it also looks perfectly clear and nice. The large red dot corresponds to the cell body of the parasite, and the purplish surface looks are representing the extension of this parasite. Following 3 weeks of house dust mite exposure, which is this common allergen we are using in mice, you can see that the number of red dots is relatively similar between the 2 conditions. However, the extent of protrusion, which has purplish surface, is dramatically reduced. And here you have the quantification because PCLS are really stable. It really, was really essential for us because we wanted to quantify this, not just providing representative picture. And PCLS allows us to really quantify this parasite coverage finally. And we saw that, for example, that the parasite number was not changing whatever the time course of house dust mite treatment or during the resolution phase. However, parasite coverage was reduced following 3 weeks of house dust mite exposure. So this is where really the inflammation is at its strongest, and it's slowly recovering during the resolution phase, but it takes at least 2 weeks to really come back to a normal situation. And what we then next looked in PCLS was whether there was any consequences of this reduced parasite coverage. And we analyzed if the red blood cell density was similar in this adventitial area comparing to following house dust mite exposure, comparing to PBS, and we had a reduction in red blood cell density, indicating that this destabilization of blood vessels, which is a consequence of the loss of parasite coverage, leads to reduced red blood cell density, and in parallel of this, an increased hypoxic area. And we have here the hypoxia quantification, where we look at if1 alpha positive CD31 on endothelial cell, and we saw that it was increased following house dust mite exposure and start to slowly go back to normal in the resolution phase. But overall, it indicates that the vasculature was damaged. There was an increased change in oxygen concentration, and that was really like basically we believe this is really not good starting point to induce like a more vasculature which is a bit more fragile. How could this work and what was the mechanism? We looked into the possible role of mast cell. As I mentioned, there is strong interaction with endothelial cell and parasite, and they were nicely present across the airway. So there are the purple cells that are lining up these airways, and they're also present in this adventitial cuff. We didn't detect massive differences in terms of air number. So all the mast cells are basically concentrated in this area in mice. Both the two types of mast cells, which I'm not going to give too much detail about them, but there are two types of different mast cells, but they are both in equal proportion in our animals, and they were not changed by even the allergen stimulation. But were they activated? That was the main question that we had. And indeed, they were activated. So what we could analyze is that you have here a mast cell body, which is here represented by the avidin staining. And when they're activated mast cell, they release these granules, and these granules are quite big structure. They're about 1 to 2 micrometers in diameter. And you can really see them. This is where PCLS was also essential for us, because we can only see them when they are in 3 dimensions. And mast cell granules, they are these sort of like tiny dots, but you can see them around the cell body. And this is indicating that the mast cell has released its granules and is degranulated. And we could quantify this degranulation of mast cell. So every time we expose the mice to the allergen, mast cells are releasing their factor. But when we stop the allergen treatment, mast cells are not degranulating anymore. And there was a nice correlation between the amount of extracellular mast cell granules, so all of these like little dots, and the reduced parasite coverage. So there was this sort of negative correlation indicating that more mast cell activated, the more parasite coverage was reduced. And this parasite retraction was dependent on the factor which is present in the mast cell granules, which is called MCP6 in mice, or the equivalent of human tryptase. And when we in vitro, we expose parasite directly to the tryptase or mast cell degranulation, we had indeed the parasite retraction, and we so identified molecularly that MCP6 was able to cleave N-cadherin on the surface of parasite, and this was destabilization of the endothelial cell parasite interaction. So that was a work that we published a couple of years ago, where we show that house dust mite stimulation induced mast cell degranulation, and these mast cell proteases, including tryptase, are inducing this loss of parasite coverage by cleaving N-cadherin on the surface of parasite, and this was then destabilizing the endothelial cell parasite interaction, leading to an increased hypoxia and reduced red blood cell density. Okay, we're doing for time. I think we're doing fine. Yes. So what I would like then after to continue is a bit what we have done with human lung vasculature using PCLS here, but also combining it with spatial transcriptomics. So when we started a bit this sort of studies, we had big problems because studying asthma in human pathology, including in children, was very difficult because you don't have a lot of biopsy. They are difficult to get. They are very small in general, so you don't get a lot of samples, and you need to generate as much data as you can from this single biopsy sometimes. And what we did is here to combine single-cell spatial transcriptomics with PCLS to get really a nice view of what's happening in asthma. So I'm just going to stop a little bit about the early life part, but I'm just going to focus on adult asthma. And we used endobronchial biopsies that were obtained by our clinical collaborator, and we performed spatial transcriptomics, here Xenium, And if you're not really familiar with the technology, it relies on this gene-specific barcode, which are going to hybridize within the tissue section a specific mRNA. And following several rounds of amplification, you can identify and analyze where is specific mRNA is present within your tissue section. So how do we get this biopsy? This is a non-bronchoscope that our clinical collaborator used. So this is from a YouTube video just to explain how this is working, but they basically use this bronchoscope and do a little pinch of the airway wall, and we can get after this biopsy for PCLS or spatial transcriptomics. And we use both samples from healthy donor and also samples from patients with severe asthma, which are all under a form of treatment, which is something to keep in mind when we analyze this tissue section. So quick overview of how the spatial transcriptomics looks like. So here you have the different biopsies that we analyzed. We have already done PCLS imaging on them. So this is after following sectioning, because these sections are about now 5 micrometer thickness. So we can do spatial on them. And you can see now the cellular segmentation, which was based on the DAPI at the time, where you can see all the DAPI staining, and now each different color here reveals a different type of cell. And the little dots that you can see now on the screen correspond to each mRNA that we can identify and associate with a specific cell type. What we did is then after for single cell spatial is that we identify which type of cells is present in the tissue, and we identified all the most normal ones that you will find in the lungs, such as ciliated cells, or the other type of epithelial cell, but we also find nice population of endothelial cell, macrophages, smooth muscle cell, fibroblast, sterol cell, and a lot of mast cells as well. And in this study that we also did, we looked into chemokine and alarmin production. And what we found is, to make a very long story short, is that in both in healthy and asthma samples, production of alarmin and chemokine were restricted to 2 main types of cells: basal cell, that we call basal cell 1, and endothelial cell 2. Both of these cell types are different state of basal cell. We don't think they are like a new subpopulation. We believe that they exist in a different state. And this state is a more pro-inflammatory one, which can secrete alarmin and chemokine. And we believe that these 2 different states coexist next to each other, and they're really important to doing normal immune response to pathogen. What was in asthma very striking, and what you can see from the difference between these 2 pictures, is that the green, the dark green cells, which are more pro-inflammatory, and the purpley one, were getting closer together. So they have this disrupted spatial architecture, and they are getting this sort of like more closely relationship between them, which we believe basically lead to this sort of increased inflammation hubs, where most cells are going to produce maybe less chemokine, but they produce more locally, so in creating this sort of like really high potent signal across this area. And so that was all good, but that was all in thin sections. So there were only 5 micrometer, which is a massive challenge when you want to understand 3D architecture of tissue. So what we did is to move toward the PCLS approach to better understand how cells were communicating together. And these are some examples of what we are currently doing using human lungs and human tissue. You have here the vasculature in green, which is all this like cellular network of cells. Mast cell in the mast cell are here in blue. You can really nicely appreciate it that comparing even to mouse model, you have about 1,000 times more mast cell, and they are present all across the tissues, human lung. They are really nicely located next to this endothelial cell and pericyte. They are present there. They are really quite nicely associated to all this structure. And they are present next to this pro-inflammatory endothelial cell, and we are currently looking into the interaction between these 2 cell types. Well, what is really good is because these human PCLS are also very stable. We can achieve really nice immunofluorescent staining. We can analyze cellular interaction in 3 dimensions. And you have here one example. For example, we can simply also count number of mast cells across different donors, which are here present. You can see there is quite a bit of heterogeneity, but overall, all type of human lung that we looked at, we identified a lot of mast cells in this tissue. They are, in general, they really like to be associated next to the large blood vessels that you can see here in the middle. That's, I guess, one pulmonary artery, and one you can nicely see the mast cells which are in blue associated next to it. So this is a bit where we are now. We are working towards a better comprehension of human lung pathology. And this is both starting from the mouse model, but also human samples where we try to combine 3D quantitative imaging with spatial transcriptomics. And this is really to better understand asthma, but also other type of allergic airway disease. And we're also making nice progress now on COPD. We're asking, there is still some questions that we're really trying to better understand, notably the impact of IL-33. Mast cells are really responsive to IL-33, so could IL-33 produced by endothelial cells be important for mast cell degranulation? We believe PCLS would be really cool to actually analyze this using live PCLS to analyze where mast cells are actually, how they are responding to IL-33 using live imaging. We're also looking into how parasite and proteases are also continue to interact, and whether we can also expand the PCLS approaches to other organs where it's already been done to some extent, but we are really trying to now export or expertise in this sort of tissue imaging for other organs. This is also a bit the summary of overall the lab where it's going. We published a bit this last year in a review where we are trying to better understand this area of the lung, this adventitial area, and analyze the interaction between the external threat coming from the air where we breathe, the role of alarmin, but also how this is impacting the local environment, such as mast cell interaction with blood vessels, and how they can impact also large type of blood vessels and the recruitment of immune cell, such as neutrophil. And on this, I would like to thank my team. So my team is mainly from with Kira, Veronia, Sanjana, and Laura that did most of the work I presented today. We are working close relationship with the team of Claire Lloyd at Imperial and our excellent team of clinical collaborators, which we're working closely with, both at Imperial, but also in the US, with Katie Cahill, Josh Boyce, and Elliot Israel at Harvard and Vanderbilt, which are really nicely shared with us some of their samples. And on this, I would like also to thank our founder, which allows me to start my lab a couple of years ago, mainly the Wellcome Trust and Actaria Foundation for funding my project. And on this, I would like to thank you for your attention, and I will be happy to answer any of the questions if there is any.
Wonderful. Thank you so much. I just have to say I'm so blown away by the imaging that you have shown of the PCLS and the bronchioles and the vessels. I'm keeping my eye here on the chat box. There are no questions entered, but I want to open it up for everybody and see if anybody had any questions. Feel free to unmute yourself and ask questions. If not, I do have one, but I want to try to temper myself and give everybody else a chance first. Does anybody else want to say something? Okay. Oh, let me just okay. I do have a question. So Dr. Joulia, you talked about using PCLS. Now, sometimes PCLS can be cryopreserved, like if it's flash frozen, but that has a lot of effects on different types of respiratory cells, right? They've been showing that some immune cells are okay. Can you talk a little bit about how it impacts the immune cells in your experience, and also the parasites and the blood vessels? I know you didn't exactly work with cryopreservation, but maybe you could tell us a little bit about that.
Yeah, so but no, that's a really important point because we have been asked before about this. I think that will really depend, as you said, of the cell type you are looking at. So we are interested into the biology of granulocytes for us. So granulocytes fundamentally they cannot be frozen. So I think we did have a lot of problems trying to snap frozen tissue because I think we're just killing most of the granulocytes that we are interested about.
Right.
Structural cell, I would think that's something to just keep in mind, but it is worth trying because some cells are more resistant than we think, especially when they are kept within their native environment. And we found it, for example, that parasites are relatively stable following snap frozen and coming back to temperature. So it all depends on the question and the cell type analyzed. But I would recommend that is also something that we currently looking at because it would be great to just be able to export frozen PCLS and send it to different labs after to see if people can reproduce data and sharing material.
Right, exactly. Exactly. And then I had a quick follow-up question, which is PCLS. I mean, the reason people want to do cryopreservation is that there's a limited window for the viability to a certain extent of a live precision cut lung slice. Can you speak to the viability in your experience between mouse and also to the human specimens? What kind of viability length have you seen? What helps or detracts from it? Maybe we can learn something from you, from your expertise.
Yeah. So the mouse PCLS are relatively stable. We found it for 2 to 3 days. They are relatively fine, kept into normal temperature and incubator type of things. But that's same thing. At the end of the day, it depends on the question asked. If looking at structural elements and responsive to injuries, that's, I believe it like 2 to 3 days is fine. Immune cell movement, we found it that 24 hours is maximum that we can look at, because immune cell movement post this is just too difficult to interpret, because it just comes to I think that just oxygen concentration becomes too high, nutrient is not right for them, so I think they become a bit more erratic and it's difficult to interpret responses. Human one, we had really big struggle on them because the human PCLS have a lot more cells than the mouse one. So they do require a lot more nutrient and oxygen, and we found it a lot less stable than the mouse one. So we believe this is maybe because maybe the human lung that we get are not inflated, or they are maybe sometimes a bit collapsed. So the concentration of cell is really high and they are not then a bit more stressed. So we found it in our experience that human PCLS more than 24 hours or 48 hours was start to be a challenge.
Oh, got it. Excellent. I don't see any other questions, so I wanted to open up one more time. And if not, I'm going to say a huge thank you, Dr. Joulia. And this webinar recording will also be posted on our website, so you can always go back and review. And if you want to be put in touch with Dr. Joulia, please let me know. Actually, Gretchen Alicia here has a question. Please, please unmute yourself and ask away.
Thank you. Beautiful work. I guess my question is, how long does the slice can be maintained? Do you always fix them and then stain? Or I thought you mentioned something about live imaging in the confocal, and how long can you then keep those slices in culture to be able to image?
That will depend on the marker. So we found it that some combination of marker works best when you stain them live PCLS when you fix them. And some marker are perfectly fine to just fix the slice, fix even the whole organ straight away, and we slice them later and stain them post-fixation, which is also perfectly fine. So that all depends on the marker you are looking at. For all the work I showed today, I think most of it is fix the whole mouse and then we slice after, and they were perfectly fine.
Okay.
So it all depends a bit more of the today. But when we look at, for example, some reporter mice work, we absolutely need to do the live imaging straight away, and that's stable. I will think for 24 hours. More than this after it starts to be quite a challenge.
Because you get a lot of background, or the tissue starts to kind of decompose?
Yeah, I mean, it's more to decompose. Yeah, it's just because the reporters start to be, basically the cells are not happy, the reporters start to be suddenly exploding, and the cells are, sometimes not really.
I see. Yeah, that's what I thought. Because I was thinking of kind of implementing this more for like metastatic outgrowth. So I was thinking more, how long could I maintain a live tissue that then is no more like what I'm getting of signal is also because the tissue is dying.
But so that's the issue that we found sometimes, is that when people is telling us, I found the hypoxia as a marker for some of my tissue, I'm like, well, yes, because of tissue is not happy. But I think we didn't really experiment much on how to improve cell viability, and I think there is a lot of really exciting work coming from bioengineering where they now trying to maintain these tissue slices for longer, providing better oxygen sensing, better oxygen availability, nutrients that are getting a bit closer to what lungs are really exposed. So I think there is a lot to be done there. It's really exciting, I think, work coming also from few labs on this. So yeah, I don't think we spend enough time to do this correctly, but yeah, I think it's
Yeah, little by little,
Yeah.
definitely. Also, I have another question, but more according to your research. I know that this was done in neonatal. Have you also planned to test this more like in older mice to see if this is maintained or they have a different, I don't know, immune response?
Yeah, that's I think something we haven't started yet. We have been asked a few times into like if we use age mice, for example,
Yes.
we do if we expose them early and after we do this live.
Mhm.
We haven't started this experiment just for lack of time, really more than anything, and also working with age mice is so expensive in the United
Yes.
Kingdom, but I try to keep a bit away from it for now. But yeah, that will be really exciting. But yeah, I think for now it's more the finance aspect, which is a bit blocking us from working with age animals.
Sounds good. Thank you.
Yeah, probably.
Thank you, Gretchen, for your wonderful questions. So with that, again, one more thank you. And also thank you to all of our attendants. Any further questions, please let all of us know. Thank you.
Yeah.
Bye-bye.
Thank you. Thank you. Bye.